right. Welcome to our August TMN Tuesday. We have a great presentation for you today, talking about combating chronic wasting disease through citizen science and how master naturalists can maybe able to assist with that. We'll get into introductions here in a minute. So just a couple of previews of what's going on in the Master Naturalist program and this webinar this morning. Just some virtual meeting room etiquette. If you're experiencing issues with WebEx, of course, if you're not on yet, you don't know that. But if you have any issues with the sound or audio, we do have a WebEx guide that can assist you on our webpage. Just a reminder, our chat function is open for on-topic discussion. Please be professional and respectful in all comments and questions in the chat box. We will moderate questions at the end of the session from the chat box. These sessions, our TMN Tuesday sessions, typically last about an hour, and you can count for the amount of time that you watched. If you missed the session, we are recording it, and if you could help communicate that to your peers who may have wanted to register for the session today too. It is recorded. It will be up on our website the following day. And you can watch our recorded TMN Tuesday webinars for AT training within the same year. And just a final reminder, our attendees are not able to unmute during this WebEx webinar. And so please keep the questions to the chat there. And as with all of our webinars that our program offers, we'd like your assistance in helping us to remain able to participate, help us to remain to offer these events in the future. And if you could take our survey here or a survey here that captures your participation, ask a couple questions. Again, it's completely voluntary. But it does help us remain eligible for the funding that supports these webinars. Okay, Mary Pearl, you want to talk about the annual meeting?
Yeah, absolutely. So a quick few announcements about our Texas Master Naturalist annual meeting that's coming up in 64 days. Definitely starting the countdown, getting closer to that event. It will be October 16th through the 19th in College Station, Texas. We have our registration live. Launched about 2 weeks ago. We have a fantastic agenda filled with almost 200 different technical and field-based sessions throughout the 4-day period of the conference. And knowing that we did have a full hotel block in our on-site hotel, we do have some external hotel block links on our website there. And Hannah can help drop that link into the chat. So if you are still looking for ways to get there to College Station and join us in October, you can find all of that information on that website there. And then a few more updates, Hannah, about the annual meeting is we're going to be highlighting some of the wonderful projects that are being submitted for our project fair during the next TMN Tuesday. And Hannah's going to be sharing more information about that on our listserv, on our website, and our social media as we get closer to our project fair presentation next month. But you can participate as a chapter and submit a chapter project into that project fair to join us as a presenter, showcasing those fantastic projects that you guys are doing in your communities, how you're doing them, ways that you're replicating, ways that you're growing your projects. Share those great ideas with other chapters around the state that might want to beg, borrow, steal a little bit of those great ideas and how to make great community impacts in conservation. So again, another link that we're going to drop in the chat for you there. But the project fair entries are due September 1st. And all of that information is also how to on our website. Last but not least, our three other contests and awards that are associated with our annual meeting also have upcoming deadlines, September 1st. That's the one day you need to remember to make it easy across all 4 of our contest and awards projects that we have associated with the annual meeting. We have our chapter advisor award, we have our video contest, and our photo art and media contest. All of that information is again available on that website. Click through those different projects, those different opportunities to showcase your work as an individual in the photo art and media contest, or your work as a chapter through the video contest and project fair, or help to support the members of your organization, your chapter advisors, by nominating them for a chapter advisor award. Looking forward to seeing everybody in October at our annual meeting.
Just a quick reminder before we move on to introductions for our speakers. If you want to be the first to know about all of the annual meeting updates, make sure you're following us on all of our socials, our Instagram and our Facebook. You can also turn on notifications for either of those if you want to. Now you will get notifications for all of our posts, and I know we post a lot, so that might be a bit much, but if you'd like notifications, you can do that. And also don't forget to sign up for our email listserv if you haven't already, and I'll drop that link in the chat in a minute as well. And when you do that, make sure you do the regular subscription so that you get emails as they come out instead of in periodic bulk emails. So if you want to be the first to know on all the new updates coming up, deadlines, all the things, follow us on socials and sign up for our email.
All right. I mentioned earlier, our presentation today is combating chronic wasting disease through citizen science, and how master naturalists may be able to help with that across the state in some certain areas. And so joining us today is Hunter Reed, and Hunter is our senior veterinarian with Texas Parks and Wildlife. He received his veterinary degree from Cornell University and his master's in public health degree from the University of Minnesota. Hunter helps the agency investigate a wide array of wildlife disease issues across Texas, in addition to assisting with wildlife health policy, rulemaking, rule development, and research. Prior to working for TPWD, Dr. Reed also worked with the Texas Animal Health Commission as a field epidemiologist, investigating disease issues in Texas livestock, primarily focusing on chronic wasting disease and cattle fever ticks. Using his varied on-the-ground knowledge of disease topics, as well as working relationships with government, academic, and private entities, Dr. Reed seeks to support the long-term conservation of wildlife resources in Texas and across the country. Also joining Dr. Reed today is Dr. Peter Larson. He is an associate professor and co-director with the Minnesota Center for Prion Research and Outreach at the Department of Veterinary and Biomedical Science, University of Minnesota. So Dr. Larson, thank you for joining us today too. And with that, I'm going to turn it over to both of you. I think Hunter's going first, and excited for y'all to be presenting with us today.
I'll actually have Peter go first, so he can share screen and I'll go after.
All right, everybody. Fantastic to be here with you today. Let me go ahead and get this going. How's that look? Is it coming through all right?
Perfect. Yep.
Fantastic. Thank you so much for having me. And I'm really thrilled to be here. What we'll be talking about today, we had this light bulb moment, Dr. Reed and I, back in January, actually. And we're pretty excited about this idea. And what I'll do in my presentation today is I'm going to go over a little bit about the biology of CWD to help us understand the disease, how it spreads, what a prion is. And then I'll talk about some new technology that we're using to help fight the spread of CWD. And just so everyone knows, I am based at the University of Minnesota. However, I got my PhD at Texas Tech. And so I know in these situations that could be a really, really good thing, depending on where you're at in Texas, or it can be a not so good thing. But I really enjoyed my time at Texas Tech. And so it's good to be here with y'all. All right, I'm going to start with this animation. This is a map that the USGS puts out and updates periodically throughout the year. And what we've done is we've stitched these together to look at an animation of the spread of CWD over time. All the light gray counties that you see in this map are where CWD has been identified in free-ranging populations, and the dots are in CWD that's been identified over time in captive facilities. So yellow means that they're depopulated. Red means, and here you can see that in this slide from 2025. Red means that they're likely current animals on the landscape. They're not depopulated yet. And so it's thought that CWD originated in North America, in Colorado. There's some varying hypotheses as to why that was. There was a research facility that was conducting research on scrapie and sheep in Fort Collins, Colorado. And the leading hypothesis is that scrapie from sheep actually crossed over into mule deer and started spreading from there sometime in the 1960s. So the biology of the disease makes this very challenging to manage, and I'm going to talk a little bit more about that. What's really important to understand is that CWD is a prion disease, and I'll highlight what a prion is. When we talk about CWD, many times you'll hear about prions, but very rarely is there opportunity to go into depth on what actually a prion is, how it becomes infectious, and so I'm just going to highlight on that real quick. Turns out all mammals have normal, healthy prion proteins. So if you had lunch, your prion proteins may actually be helping you handle some of the metal ions from your diet in your body. So these are prion proteins. These are found throughout all cells in the body. They are enriched in the nervous system, and they help regulate copper and metal ions. They also might have a mitochondrial function as well. Turns out that when this protein becomes misshapen, when it misfolds, it can actually cause disease. And so what you're seeing on the right here is a ribbon diagram that shows that protein that has adopted a different form, a different three-dimensional shape. And this is very similar to, if you've heard of amyloid beta plaques in Alzheimer's disease, that those plaques, the amyloid beta, is very, very similar to what this looks like here. So this stacked form on top of each other. So what happens is in prion diseases, the healthy protein is misfolding. It's forming these fibrils, almost like amyloid beta plaques in Alzheimer's, and those are growing, if you will, throughout the nervous system, and eventually they colonize the brain where they can start to kill neurons. I'm just going to talk a little bit more about how that happens here. So if you have a misfolded prion, let's say I'm a white-tailed deer and I've just consumed something that has these prions in it, if that were to go into my GI tract, it could start interacting with that misfolded prion, it could start interacting with these normal prions, and essentially it starts daisy chaining. So there's a domino effect that starts to occur through the animal over time. And this is actually an image of what these prion fibrils look like. And so what they do is they start growing like crystals almost. And it can take as long as about 2 years before they really start colonizing the brain, actively killing neurons. And the outcome of that is a prion disease, right? And so if this were occurring in cattle, it's BSE, in sheep, it's scrapie, in humans, it's Creutzfeldt-Jakob disease. And in deer, it's CWD. So these prions are spread both directly, animal to animal, and indirectly in the environment. And this is one of the issues that makes CWD very difficult to manage, is that these prions can actually end up in the environment, whether it's through carcasses, feces, or saliva, and they can remain infectious for many years. As I said, that process of those prions, those crystals, if you will, kind of growing through the animal, it can take upwards of 2 years, but sometimes that's faster or slower. And we see this in all prion diseases. It depends on genetics. The main thing to understand is that no deer is immune to CWD. It's 100% fatal. And in some animals, if they have a genetic background, it could spread faster through them. And others, if they have a different genetic background, it could take maybe 3 or 4 years, so it can spread slower. That whole time, though, they can be putting prions out in the environment. And then what we found on the research side of things is it takes about a bleach solution of around 50% is the only thing that we've identified to reliably destroy these prions. But that's only really effective on impermeable surfaces like metals, hard plastics, etc. So some of those unique surveillance challenges, what I'm showing you on the right here is just a model of CWD prevalence on the y-axis and time on the x. And there's these different phases, or the different colors there. And the main thing to understand from this is that you have an introduction. So you're in year 1, And over time, if the disease is sort of percolates over the first few years, and over time, it will start to enter into what we call a transition phase and then an endemic phase, where really the prevalence in the regional herds just starts to skyrocket. There's now counties in Wisconsin, unfortunately, where like 50% of bucks are positive with CWD. And so what's super challenging is that in those early phases, you can transmit. The disease can be transmitted directly or indirectly. And because it's such a slow moving disease, it can take 2-3 years to kill an animal, right? That animal can be passing it on, if you will, interacting with other deer, shedding the prions in the environment the whole time. And the problem is that there's a significant lag time with the current diagnostic tools. And so often we are, unfortunately, years behind the disease. And what we really need are new tools that help us find CWD early, so that the management strategies that Dr. Reed and others in Parks and Wildlife can utilize to help prevent spread. But it has to be implemented early on, right? And so that's our window of opportunity to deploy new technologies. That's where this technology called RT-Quick comes in. And this is something that we've been specializing on in our University of Minnesota labs really for the past about 8 years now. This is a fantastic diagnostic assay. It's been around since around 2010. It's being used increasingly for human diagnostics, not just for Creutzfeldt-Jakob disease, but it's also being used now for Parkinson's disease biomarker detection, for Alzheimer's, for ALS. And so frequently when we see new technologies like this, they are found on the human realm, but they have applications on the animal and environmental side. And that's exactly what's happening with this assay. So it's just beginning to be used more broadly for CWD diagnostics. It's still in the early phases. There's a number of research labs throughout the US, Canada, globally using this to better understand CWD biology. But what's really exciting about this test is that you can use it to identify prions in tissues. So all the tissues that are used for CWD diagnostics, we can use for surveillance, right? The existing tissues like lymph nodes and brain. But this can also be used in feces, saliva, environmental samples. And so that means it creates many exciting opportunities for CWD management. And this is just, without going too much into the nitty-gritty details of RT-Quick, this is essentially how the method works. You have your proteins. Remember those normal proteins. We can actually grow those up in the lab. And then you have your sample. Let's say this is a lymph node from a deer that you want to test, and it's positive. So what you do is you essentially mix these together in a 96-well plate, or it's akin to a test tube, like 96 little test tubes. And you basically just do a shake and bake. So you're putting that in with some reagents into this box. It's basically an incubator and a shaker. And over time, if it's positive, you will convert these normal prions into the misfolded form, and there's a little dye in there that binds to the misfolded form, right? And if you beam that with light, it will fluoresce. And so over time, if it's a positive sample, you'll see this like red curve here. That's what the sample would look like if it's positive and those prions are present, because it's emitting this light signal. If it's negative, or a sample with very little to almost no prions, it will look like this, and you will not convert, right? You will not convert those prions into the misfolded form. So how can we leverage this to fight CWD? There's lots of exciting ways, and we are using all of these methods now with various agencies throughout the US and in Canada to help fight CWD. And so you can view RT-Quick as this Swiss Army knife that agencies can leverage for different applications. We can test scrape sites. We can test the soil at scrape sites to identify if prions are being left like in the urine associated with scrape sites. Skeletal remains. This is an image of a skull that is probably about 3 or 4 years was on the landscape for about 3 or 4 years, and we could swab the inside of the brain case and find those prions and determine that, yeah, this animal was positive for CWD most likely. We can do live animal testing, and one thing that is super exciting is saliva. And this is what I'm going to focus on for the rest of the talk, is that we can leverage saliva. This could be licking branches, but also be surfaces that deer lick. We can actually look for those prions in the saliva. So from the University of Minnesota, we had made all these discoveries, new protocols for detecting CWD. We developed new technologies. And we got to a point where the demand for using some of these new protocols with RT-Quick was beyond what we could support within the university system. And so we, with the help of the university, we launched the startup company, the spinoff from the University of Minnesota, Creogen, back in 2022, really with the aim to provide folks access to these RT-Quick technologies. So in 2022, that same year that we launched the company, we made this incredible discovery. And this is collaborative research with Dr. Jason Bartz at the University of Creighton, where experimentally in his lab, they showed that you could take CW prions, you could put them onto particular surfaces and then swab those surfaces, extract the prions from the swab, and test with RT-Quick. And that was sort of this light bulb moment, because it's like, wait a second, we know that the prions are actually in saliva after a few months. What if they're licking a surface? Can we actually do that? Can we actually go and swab that surface with this new protocol and detect prions? And it turns out that we can. So this is a way we believe that we can help leverage this technology, swab saliva-based technology, to find CWD early within this critical time period early on in the stage of the disease spread. So on the Creogen side, so we've licensed this technology from the University of Minnesota. We're offering this through Creogen, and this what gets us into the next generation CWD surveillance side of things. So we know that those prions are found about 3 to 6 months after infection. They're shedding them in the saliva. And we know that the metal surfaces that deer lick can be swabbed for those prions and tested with RT-Quick. And so we've tested this now on a number of herds. And it shows great utility. Herds that are known positive and herds that are not detect. So we know that this method is working. And so we have these Prio scouts in the landscape. We're collaborating with various folks to get them out there. So what we've identified using this approach of swabbing surfaces is that we're picking up the disease at least a year before traditional methods, and we've done that on numerous occasions now. And how this works is we design this metal housing that you can just take a 4-pound mineral brick and put that. These are sold in Tractor Supply, Fleet Farm. They're really all over the place. You can load that mineral brick into this housing. And then over time, you can periodically swab the surface as the deer are trying to get to that mineral brick. We can test that and then give the results to the state agencies. And this is just a video showing a buck. This is out of South Dakota that's interacting with one of these Prio scouts. So you load that, you strap it to a tree or fence post, you can monitor activity. We think that you can actually pin down which animals are positive, like individual animals that are positive, just by using this technology, swabbing and trail cams. It's a vertical placement. We know at least 16 milliliters is required to transmit CWD. And so we designed this with the idea that we're not going to allow saliva to pool. We can bleach this periodically, it can be removed. And so this is how we're transitioning now to leverage this to help with real-time surveillance efforts. So we believe that we can leverage this Prio Scout to conduct CWD surveillance year-round. And so this gets at that real-time information that the agencies, that Dr. Reed needs to be able to help manage the disease effectively, is this real-time year-round data, right? And so this is where that light bulb moment kind of went off when talking with Dr. Reed a few months ago. Is like, this is like the perfect situation scenario for a citizen science program, because it's really difficult for agencies to be able to get access to various areas, to be able to survey like in urban, suburban settings. And also there's lots of folks that want to be proactive and help fight CWD, but there hasn't been a method really in the past for them to help do that other than testing through the normal way. And so what we're envisioning here is this will be a way that we can actively prevent and proactively prevent the spread of CWD in Texas by forming a partnership like with the Master Naturalist Program. And we think that the Prio Scouts, as I said, can detect this disease early, maybe as early as 3 months. This would be one of the earliest ways after an infection to find CWD, because the prions are actually being excreted in the saliva that early on. And that can be leveraged to help critical time. It gives the agencies time to be able to identify the disease and implement the management strategy early on, so that you're avoiding that exponential raise over time of prevalence. So together, we think we can create the world's first early warning CWD detection network, and that can help safeguard Texas deer for generations to come. That's my portion of the presentation. Thank you all for listening. I'm going to stop sharing and then we can transfer over to Dr. Reed.
Yeah, and there are a few questions in the chat. If you're able, Peter, you should be able to help answer. I know Hunter was also while Hunter's presenting. But for the most part, we're going to hold all the questions that are unanswered for the end of the session. So
Okay.
Hunter, you should be receiving the presenter privileges. And whenever you're ready. Okay, looks good.
Perfect. Good afternoon, everyone. Dr. Hunter Reed here. So I'm going to talk a little bit, and Peter gave a really good overview of exactly what this technology is, what CWD is, and I wanted to get more specific about how we are trying to implement and use this at Parks and Wildlife. And really, to preface, I kind of wanted to take some snapshots of really these maps in Texas of how CWD has really expanded across Texas, not only in free-ranging herds, but then also in captive herds. And as we had seen earlier, these gray areas where we have free-ranging detections, and these dots, yellow being a depopulated facility, red being a not depopulated captive facility. But those dots are signifying captive detections. And as you can see, in the last decade, we've had a substantial increase in the amount of both free-ranging and then, but most markedly in our captive deer detections. And that's been for a variety of different reasons, and really wanted to address those, at least for CWD epidemiology in Texas. And epidemiology is really just a study of the risk factors in spreading and increased transmission of a disease. So when we're talking about CWD in Texas, when I'm working with our big game program, as well as our biologists, we're really focusing on two different types of populations, one being a free-ranging population and one being a captive population. Both are very sizable in Texas, with around 5 million free-ranging whitetail mule deer, and then also around additional 80,000 captive whitetail deer in Texas. And both these populations can get infected through the same means. This could be through species movement of infected animals, it could be environmental contamination. With captive populations, you also have to think about contaminated feed, veterinary practices, reproductive practices. And of course, these populations are not independent of each other. This could also include fence line contact, escapes, runoff from heavy rains, scavengers, invertebrates, and more. Additionally, you have to think about the types of factors that could amplify an infection once it becomes introduced to an area. Where we have free-ranging herds that tend to be more limited in scope in their movement, typically lower densities, and then also have a larger area to distribute contamination, which is a bit counterintuitive. But compared to captive populations where you have a much higher degree of inter and intrastate movement, higher densities, in a smaller area where you can have a bunch of contamination build up in a very small area over time. This means that in captive populations, we can see the disease spread much more rapidly than we otherwise would see in free-ranging populations. And this has contributed to the spread of CWD across the nation through both of these mechanisms. When we're talking about captive deer, our department is very interested in trying to figure out ways of which we can detect the disease as soon as possible, because as you can imagine, with around 650 captive breeding facilities in Texas, around 80,000 deer, this means there's a potential for one positive facility to then transfer that, whether it be a release site or another breeder, and that could quickly tie up a lot of different herds and expose a lot of animals. So really trying to focus on increased detection or having higher sensitivity and greater probability of detection is critical to that end. And this is an example of one of the types of interconnectedness that we see. And these are our positive herds that we had in Texas up to 2023. And as you can see, they have a wide variety of historical movements between them and back to those original facilities. So this is not to say that each and every line constitutes how a facility got infected, but it does illustrate that these infected facilities have historically been very, very interconnected. And we do know that some of these connections have resulted in some facilities becoming positive. And that's just on the breeding side. We also have to be cognizant of what's going on with release sites too. And at least for captive deer populations in Texas, we have rapid dissemination of deer every spring and every fall. And this could constitute around 20 to 30,000 deer every year going out to ranches, whether they are being used to improve the genetics on a specific release site or being used as shooter bucks or put and take type of operations. So this is really just to show you how really this map is showing all the exposed release sites across Texas since 2015 when we first detected this in captive populations. So this really shows you the implications of not finding the disease in an acceptable period of time. And this is not to say that captive deer are the sole or the only deer that can move in Texas. And what I'm showing here is a free-ranging movement and a project that we actually have ongoing up in the panhandle of Texas. This is in collaboration with Texas Tech University, Cesar Claiborne Wildlife Research Institute, along with partners over at Sul Ross. But part of this is looking at how these populations of CWD susceptible species, and in this part of the world, this includes elk, mule deer, and white-tailed deer, and seeing how they move on the landscape. And as you can see, when we have these collars and movement data, and this is just from the last 3 or 4 months since they've had the collars on, that you can start to see that, yes, there are some localized movements too, but then you also have some very substantial movements, especially for elk, in addition to some preferential areas or wildlife corridors along the Canadian River. So this is to say that in certain situations, certain habitats and environments, you can have a quick movement of free-ranging animals across the landscape that can contribute to the dissemination of prions. We actually utilized RT-Quick ear biopsy punches with Peter's lab to test each of these animals following collaring. Fortunately, we didn't find any positive animals. However, we're going to track them through time and follow up with RT-Quick as well as other regulatory testing to monitor their disease status. So really, when we're looking at it from the parks and wildlife aspect, it's really why are we doing this testing? What is the point? And what are we going to do about it? Because it's really the primary difference between a monitoring program and a surveillance program. Monitoring is good just to be able to keep your finger on the pulse, but if it's not going to change any decision making, then it really doesn't constitute a surveillance program. Well, the reason that we want to keep our finger on the pulse, but also make sure it informs our decision making, is because of the consequences of this disease. One of them, probably being the most apparent, are biological consequences, is that it does kill deer. We have several instances in West Texas in mule deer and the Trans-Pecos, as well as in captive herds, where you see sick animals. They do not do well, and they ultimately die from the disease. Additionally, once you have CWD established in a population and reaches high enough prevalence, then you can start to see changes in age structure. And this means that you have populations, especially when you look at mule deer, that with this additive mortality factor, are not able to sustain themselves. And you can see decreases in the ability for people to harvest animals that are uninfected. People have concerns about eating infected animals, in addition to the fact that you might not have as many trophy animals on the landscape, which has a plethora of other effects when it comes to economic as well as human-related factors. Economic-wise, CWD is a disease that warrants management, and it's costly to manage. And it's not a problem that starts and stops and goes away. It's a constant force against those populations in their efforts to try and thrive. This also means that we have implications for hunters. Historically, we've had to develop rules that can be inconvenient for hunters as it relates to CWD management. In addition, there's additional regulations that we've had to put in place as it relates to captive deer management and movement. But there's also the human-related factors. There's CWD affected animals can pop up in people's backyards. It's distressing. They can get hit by cars, and they can presenting a safety hazard, as well as this question, or the jury's still out on the zoonotic potential of this disease. What we've seen with bovine spongiform encephalopathy, or quote unquote mad cow disease, this disease was able to cross that barrier and infect humans. With the many strains that we've been able to see with CWD across the world, it is not completely out of the question that one of these strains might be adapted to humans and ultimately cause disease. But as of right now, there has been no definitive evidence to suggest that disease has been caused by consumption of meat from these animals that are infected with CWD. And really to highlight, because of these important management implications, this has really helped develop our rules over the years. And these red boxes are really showing you the types of measures that our department has taken for at least captive deer in this situation, where we have where we've required antemortem testing before the purchase and release or transfer of any of these animals. We've also had these breeding facilities complete 100% postmortem testing, which has been upped and is at a lower age range than it was before. This has historically evolved from 20 to 50 to 80%, and now is 100%. So all of these steps have been adopted because of our increased knowledge of CWD and the importance that the department puts on managing this disease. Furthermore, it's just not captive deer where we have changed rules. What we've also done is historically we established zones, and in these zones we had required 100% postmortem testing, as well as carcass movement restrictions, essentially requiring people to quarter out animals and dispose of those and leave those infectious parts, or most infectious parts, such as the head and spine, within the zone. Because of the growing incidence of CWD in captive facilities and the increasing burden that zones place, we've had to change really our approach to surveillance in Texas, and really the rules of which that were formerly used within these zones. So as of right now, testing or postmortem testing of hunter harvested animals is voluntary. However, that does not mean the department has stopped testing deer. We've maintained those efforts. However, it has provided a little bit more convenience for hunters not having to use a check station. Additionally, we've loosened up our carcass disposal rule or carcass movement restrictions and converted them to carcass disposal rules. This was intended to provide hunters a much more flexible array of options, many of which are already being used by hunters, as well as trying to educate people on proper disposal of parts. So instead of having to quarter out your animal and leave those infectious parts in the zone, we now allow you to transport your animal out of those areas. However, your processor, and anyone or taxidermists are all required to properly dispose of those indirectly or directly into a landfill. Leave it at the site of harvest if you choose, or you can bury it at a depth of 3 feet or more below the ground. So in an effort to really, because of the changes that we've undertaken for rules governing captive and free-ranging deer, we've also wanted to take another look at our surveillance program here in Texas. And what we've tried to work on is trying to identify areas or ways that we can make sampling more efficient and maybe keep the same sampling or even increase sampling because of the fact of our transition from mandatory sampling in zones to voluntary. So what we've done is we've partnered with programs such as SOP for CWD, which is an effort led by a lab out of Cornell University in New York State, working on trying to figure out what are some attainable sampling goals here in Texas, and really trying to identify how can we most efficiently sample for the disease so that we're spotting it at a prevalence that is prior to establishment, but then also is not exceptionally taxing on the department staff and state resources. And as Peter had shown in this graph, he had shown this graph earlier, you can see that really at this initial introduction of CWD, it is extremely difficult in free-ranging populations to detect that disease and requires a huge number of samples. However, as this disease becomes more established, it becomes easier to detect. However, once you reach, at least in this graph, around 10% prevalence within an area, really the cat's out of the bag. So what our department's trying to do is figure out how we can optimize our surveillance protocol for this transition phase, where it's early enough where we can intervene on reducing the prevalence of CWD, or at least keeping it stagnant over time. But also it's late enough where we're really not getting a lot of really not so helpful samples and spending a huge time trying to find a disease at a level where it's having very little impact. And as you can see, part of this effort is going to be postmortem surveillance. We're not going to completely stop that. From our past sampling year, when we had around 14,000 samples, this forms a critical backbone of our state free-ranging deer surveillance program. However, these tests, which use ELISA and IHC, ELISA is enzyme-linked immunosorbent assay, and IHC is immunohistochemistry, they really require these two tissues, the postmortem or after death, we take the lymph nodes and ovax. Well, what we've tried to do is really start implementing RT-QuIC and PMCA. PMCA is not a test that we really discussed or Peter discussed, but it's comparable to RT-QuIC in the sense that you can detect low levels of prions through the amplification of them through a sonication or shake and bake type of test. And this can test samples, not only the postmortem samples of deer, however, we could use that in feeder surveillance, soil testing, feces, as well as in ectoparasites. So in the real importance management-wise of why we're trying to dip our toes into RT-QuIC is really, it can be explained by this graph. So as you can see, the time post-exposure, time 0 being when an animal first becomes infected, and 24 months later being when the animal is likely to expire, if it's a white-tailed deer or mule deer. And then this y-axis is showing disease stage, or essentially severity or lateness of infection. So with traditional tests where we detect this disease, we'll typically detect in probably the last third of the disease stage. While this is more of a generalization, these tests are typically later in time. And this means that there's been a significant prion load, distribution of prions in the environment, and a lot of time for infected animals to have direct contact with other susceptible species in that area. So while these tests are able to detect the disease, they do so at a later point in time, of which might not be desirable. What we're looking to do with RT-Quick is really detect an earlier stage of this disease where we have much less prion load, prion distribution, as well as exposure time, less contact with other animals within that population. And this makes it all the more feasible for our department to have a more substantial effect on stagnating the rise of CWD within a population, or in some situations, I'll discuss later, where we're not able to detect the disease any longer. So we've had quite a few projects. Our department's notoriously poor at kind of tooting our own horn, but I did want to outline a few of these projects where RT-Quick has provided, has assisted the department in, on several fronts. One of them, and I know Peter's been a part of this in his own state on a separate project as well, but RT-Quick can serve as a forensic tool. In this particular situation, we had a non-compliant captive operation. We were unable to determine just how many animals had been, there's an unreconciled inventory, and we're wondering about the disease status of these particular individuals. This was a known CWD positive site. However, we really had no sense of just what the prevalence was within that population, among other things that we're investigating within that herd. And it serves as a tool for us to be able to identify animals that were CWD positive. However, that was not with the ideal tissues, which had already degraded at that time. However, we were able to use other tissues from the animal. So it showed a proof of concept of being able to utilize this technology on a forensic or law enforcement level. Additionally, we've used this for epidemiological investigations of taxidermy facilities. In one situation in Del Rio, we had a person reach out to the department saying that they thought they had a CWD positive animal in their backyard. This was an area we weren't expecting the disease, but the animal was tested and found to be positive. We went there and we're really trying to understand how this showed up out of nowhere. What we found was that she was actually, she had a taxidermy operation right next to a rehab facility for fawns. What we found was that some of the material, the water and soil, where these heads were being essentially processed for European mounts, that was contaminated with prions. So it's likely that head came in from a site that was CWD exposed, contaminated the soil and surrounding area with prions, and then exposed those animals that had been closely associated with her operation and that she was trying to rehab. Again, this was using technology PMCA, however, very comparable to RT-Quick. Another question I've seen in the chat as well is really the concern about venison. And this has been a tool that has been available for testing whether CWD is an infected meat or not. So at least for this project, which was looking at meat from an elk that was infected, shown to be infected with CWD through postmortem testing, we tested various processed and unprocessed cuts from that animal. And what we had found was that we could detect with variable products and at different amplifications of that of the test that CWD was present in those products. Now, what was interesting was that we actually found that processing or heating these samples or meats and products made some of these prions more available, which was perhaps counterintuitive to some, where you think, oh, I might be cooking the meat and taking care of pathogens. At least for prions, this paper suggests that that's not the case. It actually increases their availability. Again, I just want to emphasize that CWD has not been shown to be transmitted to people through the consumption of contaminated meat. However, there are folks that are concerned about consuming those products and may want to use a test such as this to assess that. And then next we have here looking at different transmission pathways. We've shown that CWD can even get concentrated in the larva of these nasal bot larvae, which you might encounter if you're processing an animal, especially a free-ranging animal in Texas. So these larvae have been shown to concentrate these prions, and that those could then contaminate the environment. And then lastly, RT-QuIC has been used to show that there are alternative means of transmission. One of these, at least within this paper, was shown that fetal organs in animals that are born from CWD positive mothers can also have the tissues test positive for CWD. So a variety of uses that our department has utilized this technology, but really the question that we're all here today to ask is there a role for citizen science to contribute to CWD surveillance here in Texas? And the three areas that our department has really tried to trial this out is within San Antonio, Lubbock, as well as Del Rio. And part of the reason that we're choosing these areas has been because of pretty specific complications that we don't really encounter in other parts of Texas. Or there's specific areas where we have complicating circumstances and makes surveillance difficult to collect. So what I'm showing you here is San Antonio, and this is actually off of our public dashboard that recently went live, showing all the CWD surveillance done in Texas. And I think Michelle will be sharing that with you in the chat here soon. But really what you can see is these areas with green have varying levels of testing. The more intense the green, the lighter the green, then the more samples. The darker the green, the less samples. And as you can see, much of San Antonio, we have fairly poor surveillance. In addition, we have highly fragmented populations within these areas. So we're not able to get really uniform sampling, of which we really try to perform in other parts of Texas. So that can provide a hurdle that we need to overcome in order to increase our CWD detectability. Next, here in Del Rio, a different situation. We have very good testing and very good uniform distribution of these samples. However, what we found is that the disease has an extremely low prevalence. I should say that's the same situation for San Antonio, where we have only one detection, but very similar case here in Del Rio, where we have only 3 positives after hundreds of samples, well, thousands of samples over the last 4 years. And we also have a growing request from the public in this area to look for alternatives to targeted removal and trapping of animals and euthanasia, just to increase our surveillance in those areas. And then lastly, up here in Lubbock, additional issue that we have is that we have these wildlife corridors. So where you have very specific areas where animals are seeking to congregate, we want to make sure that we can establish a surveillance protocol on the front and perhaps in Lubbock, more northerly end and more southerly end, to see when CWD, which has been largely localized to this Ransom Canyon area, when it might escape. What you might notice here is that there's another orange dot, and what we're not expecting is really for this disease to somehow pop up in inside the loop at 289 in Lubbock. Because we hadn't detected that disease for 2 to 3 years after this initial detection. So really, we were looking for tools in which, how could we have anticipated this sooner? How could we have reduce the spread of reduce the spread of CWD from this area if possible. So as you can see, there's a variety of different situations that are not, that don't fit neat and well into our typical surveillance protocol. However, we're looking at really Peter's technology, this RT quick testing of these sentinel units, really to provide a non-lethal option, increase the efficiency of our testing, as well as use a more sensitive testing technique in certain situations, as well as be able to create an adaptable surveillance system where we can proactively and actively place these units in areas that we are concerned about the spread occurring. And if detection is made possible or detection is found, then we're able to adapt our management, targeted removal in ways that are most efficient or concentrating on the areas that we're most concerned about. And overall, this is increasing detectability, monitoring spread, increasing our sampling efficiency, and refocusing our effort in areas of greater concern. So really what I want you all to take away from this presentation is that CWD is relatively new to Texas when it was first detected in 2012. It's expanded its range across the state and localized areas fairly quickly, just within the past 10 or so years. And the main contributor to that has been animal movement. And we really need to emphasize the important impact CWD can have on Texans. It's going to affect them biologically, economically, as well as impact humans in areas that you might not expect it, just in your backyard, suburban and urban areas. And really, RT Quick can provide us the opportunity to mitigate those impacts by improving our surveillance and making our ability to find the disease in areas that are perhaps hard to sample or they're very low prevalence settings. So really wanted to put that out there to you all. Know that we're trying to pilot this right now, but in the future, we might be looking for opportunities, especially if you're in Houston or Dallas or some of these areas where we have really poor sampling, to try and provide an option where people can get involved with CWD, become aware of it, increase education, but even serve an important role surveillance-wise that's going to be helpful to them in the department. So thank you all, and if there's any questions, please let me know.
So Peter has been answering tons of questions in the chat, and actually I'm going to defer to him. Peter, do you see any that need to be still answered? Thank you
some
for
folks are
doing that.
Yeah, happy to help. And happy that some of the answers I was giving aligned with what Hunter was saying kind of in real time. So that's always good. Folks are asking how much is the Prio Scout? And right now, like if you were just to go to the website and try to purchase one, it'd be like $50 for the Scout. And then the swab test is $70, but view that as like you're testing however many animals are going to that, right? To the Prio Scout, that's how many you're testing with the swab. But the good thing is, we offer discounts in a citizen science program like this. What we would try to do from the company's perspective is we wouldn't want to charge for the Prio Scout. I think we would bring that down, that price point down, or provide it at cost. And then we would work with Parks and Wildlife and you all to lower the cost per swab test. So there are options for this going forward that we can help with. I don't know if there's other burning questions. Hunter, here's one. How would site selection work for this?
Oops. Oh, I'm still on. Okay. Yeah. So in terms of site selection, the few areas that we've focused on right now, we've really tried to get participation voluntarily from the community first. So what we've done is reached out to really local government, and they've been able to disseminate the word. In addition, and people have voluntarily come to us However, if we do have, we have been trying to place these somewhat strategically. So we have been trying to at least do some door-to-door in some of those situations as well. This is certainly a smaller scope project at this point, so we're still figuring things out. But I know that another question that had come up earlier was, well, aren't you concerned about feeding and potential transmission of this disease? And that's a very good question. Gotten it a couple of times now. And I guess from my perspective, if this was the first occurrence of feeding occurring in Del Rio, Lubbock, or San Antonio, it might be different. What I can tell you about these areas in particular, that risk factor is far very increased. There's a lot of feeding in these areas. There's already salt and mineral licks in the area. What we're really trying to do is this is not really going to be all that additive onto what's already happening. And all we're really trying to do is really look for disease that right now we know has been detected before, but might be an extremely low prevalence. And this might be the only tool that we have where we can find that in a very quick and efficient fashion.
And we designed the Creo Scout to help with this particular issue, right? Because we know, like being the scientists, we know that bringing the animals together in landscape isn't necessarily a good thing. But we designed it so that its saliva doesn't pool, so that it can be bleached, so you can swab it and then you can bleach it. And if you ever get a detection, you can just yank it out. Obviously, communicate. There'd be data to Parks and Wildlife, but really, if you can find the disease early on versus letting it percolate for years, it's a game changer, right? That's how you can really, really help fight it in new ways.
Perfect. I feel like there was a ton of questions, and I'm trying to go through the chat, but I don't know what might be the best means. I know that we're over our hour, but if you guys wanted us to answer them in some other form or fashion, just let us know. Otherwise, if people want to reach out to us, more than happy to have a chat, and you can provide our email information.
I think, Hunter, some of the questions kind of revolve around CWD 101, kind of the intro to CWD. And so is there a resource we can point those online today to kind of answer some of those questions? And then the rest we can pull from chat and get them over to you to maybe review and answer those that were asked a lot. And we can get that up on a FAQ on our website.
Yeah, no, I think probably one of the best places for information, at least as it pertains to Texas, is going to be our CWD landing page. I can certainly provide that information so people can take a look. But that's going to provide you with all the latest press releases specific to Texas, some very short videos between me, Dr. Sarah Wyckoff, talking about the disease, in addition to really providing information about if folks want to have sample kits, if they want to better understand hunter rules, responsibilities, or want to know how to report sick deer, in addition to other history. All of that information will be on the CWD landing page. And I can probably I'll copy it and put it in the chat here if folks are interested.
And it seems like there's lots of interest in some local participation, and so we can visit about what that looks like and how to distribute and moving further ahead with that.
Fantastic.
Perfect.
And I put in the chat too some it's from Minnesota, but there's one fact sheet that we get a lot of interest in, and that's the surface decontamination fact sheet, and so that's found there. So I put that in the chat as well.
Okay. We are over the hour mark, and so we'll pull the questions from this and get them over to you, and we'll be following up on where we go next.
Thank you so much.
Thank you.